In the following video, we're going to cover Archaea, from a basic overview all the way to why Archaea matter, and everything in between. So Archaea are a distinct domain of life. They're not bacteria nor a eukaryote. They possess unique adaptations that allow them to thrive in extreme environments where bacteria and eukaryotes will fail. And then what makes them unique? So here we have a comparison between bacteria and Archaea. So bacteria have peptidoglycan, while Archaea have pseudopeptidoglycan, or an S layer. Bacteria are ester-linked, Archaea are ether-linked. Bacteria are pathogenic, while no Archaea have been known to be pathogenic. Bacteria have a bilayer, while Archaea have a monolayer. And both of them have no nucleus, circle DNA, no membrane-bound organelles, and use binary fission for reproduction. Let's jump into thermophiles. Thermophiles are heat-loving Archaea. Examples include Sulfolobus, which is found in volcanic springs, Pyrococcus, which is found in deep-sea vents, and Thermococcus, which is found in hot sediment. Hot environments pose many challenges, such as high temperatures denaturing protein and melting lipid membranes. To adapt, Archaea have stable membranes, which contain ether-linked lipids and a monolayer to prevent breakdown at high temperature. Heat-resistant proteins contain more salt bridges, and hydrophobic cores help resist denaturation. Reverse DNA gyrase introduces positive supercoils, making DNA more heat-resistant, while heat shock proteins help to refold denatured proteins. These adaptations help Archaea thrive in hot environments. Now let's jump into halophiles. Halophiles are salt-loving Archaea. Examples include Halobacterium, which is found in the Dead Sea and salt flats. So challenges to a high salt environment are dehydration by osmosis and salt disrupting protein structure and enzymatic function. To adapt, Archaea have internal ion balancing. Instead of pumping salt out like bacteria, halophiles maintain high intracellular potassium and chloride concentrations to match external salinity. They also contain salt-stable proteins, which are highly acidic with negative charges that attract water and remain functional in salt. They use light-driven energy production, so Halobacterium use bacteriodopsin, a light-driven proton pump, to generate ATP. Now, let's check out acidophiles. Acidophiles are acid-loving. Examples include Sulfolobus acidocaldarius, which is found in hot acidic springs. Challenges to an acidic environment is protein denaturation and proton influx, which can kill cells. To adapt, Archaea have membrane resistance. They contain ether lipids that prevent acid penetration. They have proton pumps to active transport pumps to expel excess protons to maintain an internal pH, and they have cytoplasmic buffers, which are molecules to neutralize excess acid and protect proteins. These adaptations allow them to thrive in an acidic environment. Now let's dive into methanogens. Methanogens are Archaea that produce methane. An example is Methanobrevidbacter, which is found in the human gut, and Methanococcus, which is found in hydrothermal vents. So challenges are they live in an anaerobic environment, so there's no oxygen. To adapt, Archaea, instead of using oxygen, they convert CO2 and hydrogen to methane to generate energy. They have a unique electron transport system. They use coenzymes like F430 and methanopertin to capture electrons and drive energy production. Ecologically, methanogens play a key role in carbon cycling, breaking down organic matter. This breakdown is important for the environment. So why do archaea matter? In biotechnology, thermophilic enzyme used in PCR revolutionized molecular biology. Methanogens used in biogas production create renewable energy. In astrobiology, archaea could exist in extreme environments on Mars or Europa, which are deep sea vents under ice. For the climate, methanogens contribute to greenhouse gas emission via methane production. Understanding their metabolism could help mitigate climate change. Thank you for watching, like and subscribe.